Plant and animal breeding has long been the foundation of global food security, continuously evolving through scientific advancements. Traditional breeding approaches relied heavily on phenotypic selection, which later advanced with the advent of molecular markers and genomic selection. However, these methods often overlook the vast genetic diversity hidden within species, limiting the potential for improved traits. The introduction of pan-genome analysis into breeding strategies is revolutionizing how genetic information is utilized. A pan-genome encompasses the total genetic diversity within a species, including core genes found in all individuals and variable genes that differ among populations or subgroups. By leveraging pan-genomes, breeders can unlock untapped genetic potential, improve crop resilience, enhance livestock traits, and accelerate breeding efficiency. The utilization of pan-genomes in breeding strategies has become particularly relevant in addressing challenges posed by climate change, emerging diseases, and the growing demand for high-yield, nutrient-rich food sources. Traditional reference genomes, typically derived from a single or a few individuals, fail to capture the full genetic diversity of a species. In contrast, pan-genomes provide a more comprehensive view, enabling the identification of rare and population-specific genes that contribute to desirable traits such as drought tolerance, disease resistance, and enhanced nutritional value. This documentary explores how pan-genomes are transforming breeding strategies, delving into key applications, recent research findings, and the implications for global food security. A pan-genome consists of three major components: the core genome, the dispensable genome, and the unique genes. The core genome includes genes present in all individuals of a species, often associated with fundamental biological processes. The dispensable genome comprises genes found in some but not all individuals, typically contributing to variations in traits such as stress response and disease resistance. Unique genes are those found in a single or few individuals and often play a role in environmental adaptation. The concept of the pan-genome was first introduced in bacterial genomics but has since been extended to plants and animals. In plant breeding, the pan-genome approach has uncovered significant genetic variations that were previously overlooked. For instance, in rice, the pan-genome analysis of diverse accessions revealed structural variations affecting agronomic traits such as grain size and disease resistance. Similarly, in wheat, pan-genomic studies identified genes responsible for tolerance to extreme environmental conditions, paving the way for climate-resilient crop development. In livestock breeding, pan-genome research has provided insights into genetic variations associated with meat-quality milk production and disease resistance. The cattle pan-genome, for example, has identified breed-specific genes influencing milk composition and adaptation to different climatic conditions. In pigs, the pan-genome has revealed structural variants linked to reproductive efficiency and disease susceptibility, aiding in the development of healthier and more productive livestock populations. The application of pan-genomes in crop breeding is revolutionizing the way breeders select for desirable traits. One of the most significant contributions of pan-genomes is their role in identifying structural variations, including copy number variations, presence/absence variations, and large genomic rearrangements that influence key agronomic traits. These variations are often missed in traditional genome-wide association studies that rely on single reference genomes. A compelling example of pan-genome utility in crop breeding is its application in maize, also known as Zea maize. The maize pan-genome has revealed extensive structural variations that regulate kernel size, plant architecture, and stress tolerance. A study conducted by Hufford et al. By integrating this information into breeding programs, researchers have developed maize varieties with improved yield and resistance to biotic and abiotic stresses. Another impactful case is the application of the soybean, glycine max, pan-genome in breeding for disease resistance. Similarly, in tomato, or Solanum lycopersicum, Pan-Genome Analysis has identified rare alleles contributing to fruit quality, shelf life, and stress tolerance. These discoveries underscore the potential of pan-genomes to accelerate trait improvement in major crops. In livestock breeding, genetic diversity plays a crucial role in improving productivity, disease resistance, and adaptability. Traditional breeding methods often focus on a limited set of high-performing breeds, leading to genetic bottlenecks that reduce overall population fitness. Pan-genome analysis helps overcome this challenge by capturing the full spectrum of genetic variations across different breeds. One of the most prominent examples is the cattle pan-genome, which has revealed significant genetic variations influencing milk yield, meat quality, and disease resistance. A study by Rosen et al. By incorporating pan-genome data into genomic selection models, breeders can make more informed decisions, leading to improved livestock performance. In pigs, or Sus scrofa, pan-genome analysis has provided valuable insights into genes associated with reproductive traits and disease susceptibility. Similarly, in chickens or Gallus gallus, pan-genome studies have identified genetic variants contributing to growth rate, egg production, and disease resistance, enabling the selection of superior breeding lines. Climate change poses significant challenges to global food production, necessitating the development of climate-resilient crops and livestock. Pan-genomes play a pivotal role in this effort by uncovering genetic variations associated with stress tolerance. Drought tolerance, heat resistance, and salinity tolerance are among the key traits that can be improved using pan-genomic insights. A notable example is the development of drought-tolerant rice varieties using pan-genome data. Similarly, in wheat, pan-genome analysis has revealed structural variations influencing heat tolerance. The identification of novel alleles conferring resilience to high temperatures has led to the breeding of wheat varieties that maintain high yields under extreme weather conditions. In livestock, the adaptation of breeds to changing climates is a growing concern. The pan-genome of indigenous cattle breeds, such as the African Nguni and Indian Gyr, has identified genes associated with heat tolerance and disease resistance. By incorporating these genetic adaptations into commercial breeds, breeders can develop livestock populations capable of thriving in diverse environmental conditions. Despite the significant advancements in pan-genome research, several challenges remain in its widespread application to breeding strategies. The high cost and complexity of sequencing multiple genomes pose a major barrier to large-scale implementation. Additionally, the computational demands of analyzing pangenomic data require advanced bioinformatics tools and expertise, limiting accessibility for many breeding programs. Another challenge is the integration of pangenome information into traditional breeding pipelines. Most breeding programs rely on single reference genomes for selection, necessitating a paradigm shift toward multi-genome approaches. The development of user-friendly computational platforms for pan-genome analysis will be crucial in bridging this gap. Looking ahead, the future of pan-genome research holds immense promise. Advances in long-read sequencing technologies, such as PacBio and Oxford Nanopore, are improving the accuracy of structural variation detection, enabling more comprehensive pan-genome construction. Additionally, the application of artificial intelligence and machine learning in pan-genome analysis is enhancing the ability to predict trait-associated variations with greater precision. The utilization of pan-genomes in breeding strategies marks a paradigm shift in modern agriculture. By capturing the full genetic diversity within species, pan-genomes provide unprecedented insights into trait variability, enabling the development of superior crops and livestock. The identification of structural variations influencing disease resistance, stress tolerance, and productivity has already led to significant improvements in breeding efficiency. As sequencing technologies continue to advance and computational tools become more accessible, the integration of pan-genomes into breeding programs will become increasingly feasible. In the face of global challenges such as climate change, food insecurity, and emerging diseases, pan-genomes offer a powerful tool for sustainable agricultural development. The ability to harness hidden genetic potential through pan-genomic analysis will not only enhance crop and livestock resilience, but also ensure food security for future generations. The continued exploration of pan-genomes will undoubtedly drive innovation in breeding strategies, shaping the future of global agriculture.